welcome to my YouTube channel. Today we want to look at biology. That is biology, the paper that the candidates did during their NEC exams. That is Kcse 2025. That is the paper that we are going to look at, so that we revise for you to at least try to be concerned about this year exams, so that you prepare well for paper 3 in the year 2026. All right, biology paper 3. So if you like it, you subscribe it and just share to friends, those friends that you know. May they share this kind of video to them, so that they are able also to learn from the paper 3. And paper 3 usually gives us a lot of marks, and it depends on a student who is very serious during this examination. And I think a student is must pass paper 3. Must pass paper 3. So let us start it. So the paper was very simple, and it was much of theory, and if it's much of theory, it means a student must study very well in theory. Now I should give the specimen E. Should give the name of specimen E. So this specimen E here was called Irish potato. This was Irish potato here. It was Irish potato. This is Irish potato. So that was Irish potato. A student was given that Irish potato. He was also provided hydrogen peroxide, a beaker, a scalpel, 2 test tube A and B, a dropper, a source of heat, white tile, a wooden splint. Procedure. So there was a procedure here. Peel portion of specimen E using the scalpel, and cut 2 equal portions about 1 centimeter each. Then the second procedure, macerate. You have to understand here. You macerate or cut into small pieces. Match 1 portion on a white tile and place the pieces, of course, in the test tube labeled A. Then the third one is boil the other portion in a beaker, macerate on a white tile and transfer it into the test tube B. Then this one is just boiling. And the third one is to each test tube add 3 drops of hydrogen peroxide and record to observation. Now, the observation is very much important for any student who wants to pass. That's a skill in any kind of in any in practicals. Now, in test tube A, we have been told from that is a procedure that we are supposed to put a small pieces of a portion in test tube A. Then you're supposed to add hydrogen peroxide. Now, there is a question that concerns how detoxification takes place in a liver of a human being, because that's where hydrogen is broken down to less toxic substances like water and oxygen. And simply, when you are using small pieces of Irish potato, you are macerating it. It means you are increasing the surface area. And the moment you are increasing the surface area, the rate of reaction takes place very fast. So what happens in Irish potato or in an animal tissue or a plant tissue, there is an enzyme called Kcse enzyme. So here we are testing an enzyme called Kcse enzyme. So this Kcse enzyme is very much important because the one that help to break down the hydrogen peroxide into water and what? And oxygen. So Kcse enzyme is found in both animal and in plants. Now, in test tube A, what will happen is that there will be frothing. There will be frothing. There will be frothing. There will be frothing. Stroke, we call it bubbling. There will be bubbling or hissing. Somebody maybe he had a hissing sound which was produced. Now, what do we conclude? What do we conclude when we are seeing frothing or a bubbling or a hissing sound? It can be a hissing sound. A hissing sound. Then what do we conclude when we are having that one? What do we conclude if you are hearing the hissing sound? You are using all the senses. If you are seeing some bubbling, what does it mean? That one means that there is, or somebody can say, effervescence here. There was effervescence. Yeah, right. There was effervescence because effervescence is the same as bubbling. So that one is to mean that there was there was a there was a breakdown of hydrogen peroxide by some Kcse into water and oxygen. So the oxygen which was produced is the one that you see it in the experiment. You see some bubbles coming out. You hear some hissing sound because there's production of that gas. There is effervescence because now that particular Irish potato has been macerated, increased its surface area, and the reaction is taking place very fast by a Kcse enzyme, and a gas is produced. So we conclude that because of that, we conclude that if there is bubbling and there was any, so you conclude there's production of oxygen. There's production of oxygen. Hapa, you can say there is some production. There is some production. There is production of oxygen because you cannot see bubbling if there is no production of some oxygen. So that's bubbling, production of oxygen, the one that you see in bubbling. So in B, it was boiled. You can see there is boiling. So the moment, remember, the enzyme Kcse is active at optimum temperatures. But when there is high temperatures, you are going to denature the enzyme Kcse, and there will be no broken down of what? Hydrogen peroxide into water and oxygen. And remember, if there is low temperatures, also you are affecting the enzyme. Also, it becomes inactive, and therefore there will be either low or no essence, or there will be no production of that oxygen. So if there is there was boiling in test tube B, then it means that the temperatures were high. And remember, when temperatures are high, they denature the catalase, this catalase enzyme. And therefore, what we are expecting in B is that there is no effervescence. There is no effervescence. Or there was no effervescence. There is no effervescence, or you can say there is no, frothing, okay, or you can say there is no fizzing, okay, so that's imekubalika. So all those there is no bubbling. Why do you think there is no bubbling? There is no fizzing, there's nothing happens. What does that mean? It means that due to high temperatures, or due to boiling, it denatures the enzyme. It denatures the enzyme, and the moment it denatures that enzyme, catalase, there will be no fizzing. You see, there will be no fizzing in that particular experiment. Now, question number B. You are asked, state the main or the aim of the experiment. Why do we boil the Irish potato, and also the other experiment, you don't boil it, but you're using hydrogen peroxide? So that means this experiment is to investigate. So a learner must write that is to investigate the effects, the effect of temperature. The effect of temperature on a catalase enzyme, That is to investigate the effect of the temperature on the catalase. Very, very, very true. Because the moment you put a catalase in high temperatures, they are denatured. Low temperature, they are inactivated. That's the aim of the experiment. Question C. Give the reason for macerating specimen E before adding hydrogen peroxide. We macerate, or we cut the Irish potato into small pieces so that to increase, this simply just to increase, this one is to increase the surface area, as simple as like that, to increase the surface area for of course for enzyme or enzymatic reaction, to take place. Simply just to increase surface area for enzymatic reaction. So the reaction takes place very fast. Name the organ in human body where the reaction in the test tube A occurs. Usually in human beings, this liver is where this kind of a reaction takes place. And a liver must start with a capital letter. Must start with capital letter. If there is, you can reject liver. There is somebody who writes something like that. So if there is wrong spellings, it becomes wrong. Roman 2. What is the significance of reaction to the body? State the significance of the reaction to the human body. Now, we have the reaction. And what the reaction takes place here is normally detoxification, whereby toxic substances like hydrogen peroxide in the liver, they are broken down by catalase enzyme into non-toxic or less toxic substances like water and oxygen. Now, the significant is that you can say it breaks down toxic substances. It breaks down toxic substances into less toxic substances. Yeah. So that's what we call majorly detoxification. Remember that? Detoxification. So detoxification is the process by which toxic substances are being broken down into less toxic substances in the body. Remember, toxic substances are much dangerous in our bodies. Now, suggest two other factors that are likely to affect the rate of reaction to test tube A. So test tube A, we try to cut it into small pieces. That's what one factor to increase the surface area. Other factors that can affect that, we can say number one, we can have the pH of the medium. That one is very, very much essential. We can have number two, size of the substances. Yeah, if they are very small, the way we say they are up there, we can talk of concentration. We can talk of concentration, or as well, yeah, those are the things most of the time. Yeah, those are things we can look at. pH, we can use the size of the substances, or we can look at the concentration. Okay. So let's continue. So that was question A. Now question A is over. So that's how the question basically was being asked, and was just very simple, direct to the answer. So a good student was to mention out clearly what basically he or she asked on that question number one. Now we have question number two. If you look at question number two here, you are provided with the materials and photographs. So question two, you are given some materials and some specimen. So for specimen E, I think this was it was given as I think, was it a bin? Yeah, it was a bin, Sydney. Yeah. Then J was a locust. Yeah, J was given a locust. Now, you are given two things here. Other specimen, and you're given the photographs. You're given the specimens and you're given the photographs. So the question two was given like that. So you have the specimen here, K, L, and M, being seedling, and also the locust. Now, question A. Complete the following table by classifying the specimen H, J, and M based on the observable features. Remember, the word here is observe. What you observe, those features that you observe in that particular specimen, that's what you are asked for. So if you start with H, at the question. H was a bean seedling. H was a bean seedling. So we are expecting it, that bean seedling, to be, so a bean seedling dicotyledonine. It is in a class of dicotyledonine. So dicotyledonine, that is the class. Give a reason why it is in dicotyledonine. So that particular plant was given, it had, you could have seen the taproot. Remember, we are talking of observable features, what we are seeing. So taproot was given there, and you could see some kind of broad lamina. Yeah, you could have seen something of network veins. Okay, yeah, can have network veins like that. So those are the reasons why we call it, or we classify it in class dicotyledonine. Now J. Now if you look at J, was a locust. So J being a locust, we call it class insecta. Class insecta. And remember the spelling. See, if you study small letter, then it becomes wrong. So we have class insecta. Why do we say grasshopper? It is in class insecta. Then if you look at that grasshopper very well, if someone had to see the grasshopper, you could have seen the three parts, three pairs of legs. You could have seen three pairs of legs. They are there, three pairs of legs. You could have seen it is divided into three. You could have seen divided into three, and that is very, very much important. So you look at it, three body parts, gonna three body parts. Also, that one must be clearly stated. Three body parts. The thorax, we have the thorax, the abdomen, and we have the head, the other part. So three body parts. You could have seen three body parts. You could have seen a pair of antenna. Antenna. A pair of antenna. That's also an answer for that. You could have seen a pair of eyes. That's why it is in class insecta. Then we have photograph M. Photograph M, if you look at it, is an eagle. This is an eagle. So if it is an eagle, then we are talking of class Aves. Class Aves. So why do you saying class Aves? There is some presence of what? The presence of feathers. Yeah. So those feathers is covering this feather, is covering itself. Then there is some presence of also peak. Okay. The peak. Yeah. So that's what you can see. Even you can see the scales on the legs, but also that's also very, very essential. Yeah. Then question B, explain the ecological significance of kingdom to which specimen H belongs. Now specimen H, look at the specimen H. H is a bean seedling. Ecological significance. So in where this particular plant is situated, what is the significance it brings that particular habitat? So number one, it produces or they are producers. Let's say they are producers because they can photosynthesize. They can photosynthesize. Synthesize. Okay. Producer, they can photosynthesize. That is yeah, they can photosynthesize. After photosynthesis, those are sugars and such to produce carbon 4 oxide. Okay, using using carbon 4 oxide, which is removed from atmosphere, which is removed from atmosphere. Is removed from the atmosphere. Okay. At the same time, it can respire. They can respire. Give out oxygen. Useful for animals. Useful for animals. Yeah. As much as this particular plant do photosynthesis, making of food, they remove. Yeah, not remove. Sorry, they don't remove that carbon dioxide, but they right. It is very correct. Yeah, it is very correct. Remove this carbon dioxide as much they utilize it. It is removed from atmosphere because now the animals can't use carbon dioxide because carbon dioxide is a is a, is a is a waste product in animals. So it is removed and utilized in the plant to make food. At the same time, that plant removes oxygen, which is also a waste product according to plants. But in animals, it becomes useful because they use that oxygen in breathing in gaseous exchange. Yeah, that's how it is very, very essential. Very essential. Now, part C. You are asked now, make a label diagram of a dorsal view, okay, of the abdomen of a dorsal view abdomen of specimen J. Specimen J, remember, it was a locust. Remember, this is a locust. So, how do you draw it? So, this was very much important for, so they were the skill that they